back at the world of innovative potential, and I'm rather quite excited and cheeky tonight because it is one of those like, when you're stuck and it just sort of like comes to you and it's just like the walls break and you're just like, oh my god, it works. The walls don't break. It's just more of just me. It's more of me. I love the disappointment. No, it is just me. But tonight we're going to talk about our, we did a drug development study tool that kind of got developed, and I'm going to talk about it, and I'm super excited actually to talk about it, and I'm going to talk about it right now. So, kind of what this is about. This goes really in line with the wheel of asset creation. The wheel. This was sort of one of the main drivers behind it. It's like the fifth wheel. Kind of the fifth wheel that was going on there. And basically, what's kind of happening with that. Where do you start? Like, oh my god, where do you start? It's like, stop. We're gonna cool it off to the same beats here. Change them up a little bit. Merry Christmas. Basically, what is this? Prescription development. It's a drug development cycle. It's a drug development cycle. It's a drug development wheel. Drugs, pharmaceuticals, prescriptions, molecules, miracles, and what ails you almost, basically. So I'm gonna cool this off. That's what it is. It's the cold light, not the warm light. Forgive me. There we go. The warm light. I like the warm light. It's nicer. It's nicer that way. It takes the edge off. It's not as much as a cold clinical science scapula, not scalpel, but a scapula type of mentality or a mindset, but it is sort of, this is what it is. This is sort of like the fourth and a half wheel of innovative potential. We have asset creation, we have cancer biology, we have health literacy, profit, and now prescription development. I was going to call it drug development, but there's sort of like that strung out sedative type of first impression, and we're gonna clinish it up a little bit, I guess, because this is basically covering the full sort of development cycle for a molecule. So now I got put this one. So basically what we're talking about here is I need something to fidget with, otherwise my excitement will just sort of play through, so pardon that. Back on track, 3 minutes in, and we're off to a good start. So now let's begin. The prescription development wheel from Innovative Potential Incorporated is broken down again into 4 sections. So this is what publishing on the fly does for you. It's just basically fix your spelling mistakes, get it all up and running, chase the demons away, and you're live. So it's live recorded television. So anyway, the prescription development wheel, the drug development wheel, is broken down into 4 sections again. It's in silico, in vitro, in vivo, and in clinico. We're just like, going off of those like pseudo-Latin type of things. But what's going on here is it's describing and walking through these like major stages of work. So major sort of like milestones of work, like how do you send? It's like, okay, we just did this. What is our next thing to do? This is the next thing to do, and and I'm teaching you how to do this independently. So basically, what this type of image does is it says, okay, let's start with the disease. It's a general thing. So it's like you start with the disease, and now it's like, well, any disease has to fit into this wheel to make it generalizable enough to be valuable. This is what I went through for the Joey. I'm talking to my history self here, but in the future, if I wanted to do an antibiotic will still function in the same way. And maybe today our example is going to be an antibiotic. But basically, there's sort of two ways to do drug development. One is, here's the disease, cure the disease. That's the way this is set up. Here's the disease, go cure the disease. And then there's another one that says, oh, we've had these molecules, and this is sort of only happened in the last century or so, more than the last 25 years. That's like, okay, now that we have all of these drugs made and manufactured, we know that this drug affects this type of side effect, but if we use that side effect someplace else in this disease scenario, then we can kind of we find a new use for the same drug without having to do all of that work. And that's kind of, I'm kind of in my own personal drug development life, I'm kind of in between those two, where it's just like, yeah, we got a pile of drugs that we already have, let's find a new application for those molecules and those formulations. So it's like, we got a mountain, let's make best use of it. So the idea here is, so you pick your disease, you sort of start at the top conceptually, and then you just follow the direction of it, and it leads you back to the start. So the idea is, you have your disease profile, you have your mechanism, plausibility, and this is, I just like all the other wheels, I'm gonna jump into this again. Your disease profile, mechanism, plausibility, functional group screening, analog profiles, synthesis pathways, analytical methodologies, testing protocols, physicochemical behavior, bioassay and statistics. Then you have your cellular models, subcellular, non-cellular models, subcellular models, cellular models, tissue models, translational study, little combo breaker there, and then back to animal models. And you have your ethics approval, your statistical power assessments, animal husbandry, treatment data analysis, and finally to take you back through the clinics, you have study design approval, study recruitment, treatment administration, data analysis, and information reporting. Back to the disease profile. So you right off the bat, you kind of see like this, there is a repeating pattern within these 4 sections, and a lot of it has to do with planning. First off, planning what you want to do, making sure that you have your mathematics set up properly before you do any of the work. It's very much, you're building a domino thing to just knock it down and go, but it's, here's something, this something has to work this way. If it doesn't work this way, here are some of the contingencies, because we're only going to get like one opportunity to do this once. And we have to get all of the information that is possible sort of captured at once. And then you have to do it for an entirely different set of circumstances. So a car, you might be an auto mechanic, but you're not an airplane mechanic, and you're not a rocket ship mechanic. You know what I mean? So that's kind of how this changes. It goes from being a bike mechanic to a car mechanic to an airplane mechanic to being one of the jet propulsion engineers at NASA. And that's kind of how this sort of like, that's how you can gauge the difference in complexity levels between each of the 4 sections. So that's what we're diving into today. And not that it's complicated, but it is one of those things where it's just like planning. This is to help you know what you're planning, and if you're just sort of starting out, it's like knowing that this picture exists is exactly what makes life like not so horrible. So basically, your disease profile, you have a disease, you might be a doctor, and this is sort of coming from it can come from any direction. You can like Google, Wikipedia, you could be a doctor, you could be yourself, you could just be like interested in this type of stuff. But it's like the disease has a disease or a condition or a state or an infection or some type of thing that needs to be corrected or fixed has a known type of like pattern of presentation. So, first, if it gets stung by a mosquito and it turns into malaria, bitten by a mosquito, whatever, it's going to follow the same progression pathway. If you scrape your leg and then it turns into infection, it's going to follow the same type of disease progression. If if your DNA gets a mutation and it turns into a cancer, it's going to follow the wheel of cancer biology. So it's like, for the sake of this for the sake of this video right now, this stream, the wheel of cancer biology is one disease profile, right? And it's only a partial disease profile. It actually has, a larger clinical presentation type of stuff. So there's the clinical disease presentation, and then there is what's happening inside the cells. And we're talking about the inside the cells type of thing, because you want to stop it from like proliferating. So it's like, well, you got an infection, but it's going to present as a cut with pus and an abscess in a veterinary or a hospital situation. Mechanistically, that's why this wheel goes right into the mechanism. You're going right into the microscopic world here and subcellular world right off the bat, because it's like, well, you're never going to know that the abscess is caused by bacteria. You just know that, you stepped on a rock, it got ingrained and then abscessed, and you had to, drain the goat's leg with salt water. That's sort of the idea that's going on here. Like, there's a clinical difference, and then there's like, this is all of the like rest of everything we know. The in silico world is like, it's not lab on a chip type of stuff. It's like, this is where you do all your research information. This is if you can run simulations, if you can do mathematics, if you can use a computer. This is where you use your computer, and there's a lot of information. And this is the type of information you want for drug development, personally. So the mechanism plausibility. So you have to actually sit down and you have to use your wheel of asset creation here to do your research and figure out, it's like, okay, well, I need something. I have to do a lot of research to do it. Because if your mechanism ends up being like really, it has to be like believable, like not even believable, it has to be plausible. So it's like, well, I could believe this works, but it's like, you got to dig into the nitty gritty all the way down to the point where if we do this and you come in on this and it just keeps going and it reaches the point where the thing that started the infection or whatever stops existing, then you can come back out. And when you come back out, you're curing the disease at the same time. So if you take a molecule, and this molecule can kill bacteria, and we can get it to the point where the bacteria and the abscess stop existing, then the thing can come back out. So this is the first sort of like, okay, that's how this works. That's sort of really the first stage of in silico, where you're sitting down researching stuff. What are cells? This is like like there's this huge section here is basically, okay, what are cells? What's a functional group? What's an enol? What's like how does the bacteria interact with the drug? There's multiple kinds of bacteria. There's multiple shapes of bacteria. There's multiple. I'm going gram positive and negative here, but it's like there's multiple different sort of defense mechanisms that the bacteria has that will just shut off your drug. And then you have to worry about what the body's doing on top of that. So like it goes wide and it goes deep, all at the same time here. But when you can get some idea of how is this thing going to go away, and how is this thing that I have going to make something else go away? Now you're going into functional group screening. You start shifting into functional group screening. And what I'm talking about here is the functional groups of the molecules that will be interacting with your could be a bacteria, could be a virus, could be a defective protein that's continuously synthesized. It could be a congenital thing. It could be genetic. It could be post-translational. So the gene is okay, but whatever is happening in the machinery, it just keeps, doing whatever. So there's, it could be there's a lot that goes on here for a general statement. So, but the point is, when you have the idea to the part where you can go in and you can go in and you can get out. It's like you're going on a vacation, but you got to come back eventually. You have to come back from the vacation before you move on to functional group screening. That's kind of what the deal is there. You got to go in, you got to have the fun in the sun, and you got to come back out. So functional group screening is there's a lot into the receptor molecule interaction. So there are huge data, and then this is where it's like, okay, well, this is where the huge databases are coming from, and the receptor molecule stuff is for me anyway, a lot more into the non-oncological world for me. I've spent a lot of time to sort of like hyper focus on this type of stuff, so pulling myself back out was a good idea. So what's happening here is it's like a lock and key. A lot of the biological locks and keys are very well known. They've already been really well described. They have 3D figures, they have 3D models. The online databases are extremely powerful. This is where like big data is actually so valuable, because you can know already. Here's my proposed molecule. Here are my proposed functional groups, and it's going to interact with this target. So what I kind of mean by mechanism plausibility, I'm going to go back to, so one of the breast cancer, a lot of breast cancers are driven by receptor activations, the estrogen receptor activations. So how do you make a drug interact with the receptor? Because if you turn the receptor down, it stops fueling cell proliferation effects. And I gotta watch myself on this one. Take this with a grain of salt. It turns down. So basically, I'm gonna use a more general example because it'll be easier to cram everything into an hour. So basically, functional group screening is if we change one of the functional groups on this molecule, it will interact better with this receptor. That's what I'm getting at here. So once you kind of get an idea of like how you want the shape of the molecule to look so that it interacts properly or the most effectively or efficiently with the target site. So the mechanism plausibility here is that, well, if we turn down receptor overexpression or something like that in a general case, like a general example, then the effect won't be as pronounced. But how do we get a molecule to interact with that receptor? How do we make our key fit better with the lock? So it's basically like locks and enzymes. So keys and enzymes and locks and chemicals are kind of like used interchangeably here. But the idea still kind of holds true that like it's like this and this, and it, like the molecule can do this, the molecule can do this. There's lots of configuration changes that make the body work, and that's the whole sort of ideology behind pharmacology. That it's like, okay, well, let's use this for a beneficial effect, or let's learn and understand how these systems start interacting with each other. So that's sort of like almost medicine 101. How do they work and interact with each other? But while you're doing functional group screening, look at the analogs. So it's like, well, if this molecule is this and it's interacting, look at some of the other different shapes that are going to interact with it also, because you might get something that is more or less, that's called a binding affinity, where the enzyme is going to like either sort of like be loose, or it's going to latch on and never let it go because you changed a methyl group to a chloro group. So that's kind of used in a lot of sort of pesticide development, where it's a functional group change like that can change the ability of your molecule to get to the target site, the ability of the molecule to dissolve within the body, like not dissolve, but it can change the pH and pKa properties of the molecule. It can change its ability to dissolve in fats and be lipophilic. So there's a ton of stuff to look at. And this is difficult for me because I'm actually describing molecular shapes that I've seen in the past that I've had to sit down and say, okay, well, I want a sugar molecule in acetaminophen so it gets uptaken into the tumor cell better, so that I can see whether or not this interaction is actually going to fly. Or put it on a benzoquinone. Can you get sugar in benzoquinone and put that into a cancer in vitro experiment to see if it's more effective? So that's some of this. That's the type of stuff that we're gonna get to, like in the in vitro and in vivo sections. But the idea here is like, what molecule is going to fix your problem? So that's the analog profiles, because it's like learning and expanding your reach around the basic shapes sort of says, okay, well, the one shape that you want might not be available. It might be an extremely difficult thing, but the analog will work. But an analog or an isomer or something will work just as good, and it'll be cheaper, faster to make. So that's kind of where the synthesis pathways is. It's like, even though you're in silico still, it's like, well, you still got to pull it out of thin air. You gotta know what you're doing. So synthesis pathways. This is like, how do you make the drug? Where does the drug come from? You have a molecule. It might not be an actual drug. It might not be something you can pull off the shelf. This is your university laboratory chemistry department organic synthesis specialist. So these are the people who know how, like, they end up working at like DuPont and stuff like that. So you have to really know what you want and how to make this type of stuff. But planning out how to make what you want specifically, or answer the question, where is this going to come from? And that's sort of where it is, because it might not be, it might be bioextractable. You might be able to farm this thing, and that's like, okay, it's farmable. Your starting materials are farmable. Okay, we can do this. We can do a plant extract. We can grow a couple of plants and pull out an extract and go from there. So that's sort of the idea here. So once you know how to make it, your next step is to know how to detect it. So they kind of go hand in hand, the synthesis pathways and the analytical methodologies. Really, because if it changes, if the molecule changes during the synthesis, so if you overcook your molecule, it will polymerize and you won't get it, or you'll cut your yields down, or you won't be able. You're just not gonna. You're not gonna be able to do what you want to do. So that's really like this wheel is a gigantic wheel of disappointment. You know, you know what I mean. That being said, it's like if you sit down, you take the time, you show up and say, okay, well, here's this piece and this piece. This wheel is about planning as it is disappointment, because here is something that looks legitimate to help somebody in a clinic. So the in silico part, this is kind of where and why you have COVID vaccines within a year, because there is so much work that is able to be done in silico now, that a lot of the more expensive in vivo and in vitro work that used to be done has been like, no, you sit down and you read it or you type it in. Here's what this protein would look like, or here's like, and that's an extreme example, but for like average university grad students or just sort of people like interested, you can sit down, you can Google the databases and begin to understand what, why, and how is this plausible. So some small molecules won't exist because they react with water and you can't have it. So, you might have a disease profile, you might have an idea that uses like sodium, but sodium would burn a hole in your patient, so sodium doesn't get used. So it is a little bit of that, like, you gotta at least like smarten up beyond high school chemistry a little bit. So, once you get past to that understanding there, so once you deal with that, the next part of everything, if you haven't gotten bored yet, because this is one of like the cancer bio ones that are just sort of like, there we go. Testing protocol. So what are you testing here? And it's like, you're testing your analytical methodology. So basically you have a molecule, but now it's like, well, if you're going to cure disease, it's got to go to a person, or it's got to go to an animal, or it's got to go to a plant, or it's even for a lot of this, it still applies to pesticides. So this can be even like pesticide development if you wanted to. It's just like, even I would say cosmeceuticals. Can't even pronounce that word right now. But that's the basic idea here, where cosmeceuticals, but they're cosmetics and pharmaceuticals. So if you have creams with lotions and stuff like that, your testing protocol. So basically, are you going to be this sort of like where it's like, well, what formulation is this going to use? Because you know that the body has different pHs inside of it and outside of it. So basically how this works is you have to use your analytical methodologies and your testing protocols to make sure that you know something about the would-be drug, about the molecule as it behaves in like water and like that's where physicochemical behavior shows up, where it's like, can this thing that we know it can be synthesized, but is it going to be air stable? Is it going to be UV stable? Is it going to be water stable? Do I have to, liofilize it? Do I have to make it more lipophilic so that it doesn't degrade? Is it something that can, it's like, it's really like, there's a thousand, like this is formulation expertise here. This whole, analytical methodologies, testing protocols, and chemical behavior is saying that, okay, we have a shape, we have a bunch of sticks, we made that bunch of sticks and lines and whatever, and then that has now become something we can use, but we got to know some more information about it, and then it's like, well, how are we going to know more information about it? Because everything's basically a white crystalline powder, unless it's not. So a lot of dyes that are used regularly also follow through this. Like, how do you even know that if you're in dye synthesis or manufacturing or something like that, how do you know a new dye is going to be stable? Well, you start testing it. The testing protocols are unique to what you want to find out, but it starts setting up the mindset and starts setting up what is required to do the next 3/4 of the wheel here. So you know what molecule you want, and you know what thing you want, and you know something else what you want, and what else? What else? Like, you clearly know what you want by this point, because it's either going to be able to be used, or it's not going to be able to be used. And you'd have to go all the way, like, this is one of those like game of troubles where it's just like, no, you go back to start every single time. It's like so you got your functional group screening, go back to start. You got your analog profile, go back to start. You know what I mean? Like, you got your analytical, no, you go back to start. You start at the start every single time, and you just sort of like move the next one, and then you go back to start, because there are so many failures. There's so many failures along the way, and they're all in silico too. You're not actually on a bench or laptop yet until you know what you're doing, and by the time you get to where you want to go, you have a really good idea. It's like, okay, this much, like, how to, you're gonna have it in a bottle someplace. You're gonna have, your thing in a bottle, and that is the molecule you're gonna use. That's your next wonder drug. You've either purified it, refined it, synthesized it, extracted it, and it's sitting in a bottle in the fridge in the laboratory, and you're ready to use it. But that takes us to the bioassay and the statistics. The statistics are the statistics are good. You have to be able to understand a little bit more about the results you want. So what you're doing here is you're going out beyond an action and then pulling it back in. So you're going to sit down, you're going to plan out, so it's a big if statement here. So if I do this and create a whole bunch of numbers, these numbers are going to behave, it's number prediction. So if these numbers are going to behave this way, or it's like, if this event is going to behave the way I think it's going to behave, these numbers will come out this way, and that will give me the value that I want. It's going to help me at the next piece. So you're going out one part, but then you're going out, like, if this was a train, you're starting at the second car from the caboose kind of thing, and then you're going up you're skipping 2 cars and then coming back 1. So you're going 2 steps forward, 1 step No, you're going 2 steps forward with one foot, and then 1 step forward with the other foot. And that's why this is so complicated, awkward, and usually just like, why am I stupid kind of like thing happening here. So what you're doing is you're setting up your results tables. You're setting up the information that you want that's going to back you and support you. So when you're backed and supported by your things, it means you didn't screw up, or you're able to make the actual prediction based on, if we have this amount of drug in this volume of stuff, this concentration behaves the exact same way every time. So you end up with everything in triplicates, and they're all like the exact same point, right? So that's kind of what's going on with the bioassay statistics. It's getting everything set up so that you just have to plunk in the numbers that the real world created for you. But this is part of the in vitro part, because it usually just typically applies to it. It goes further. It, like, you can bioassay a clinical trial if you want to. It's the structure of how you're going to do it, and there are books on there. There's one by J.J. Hubert. He was my teacher, not once, not twice, but thrice. Go check it out. It's called Bioassay. It's an advanced book. It starts, I think, second or third year statistics course. So it's like a sophomore or junior level course. So basically, once you figure out what you want, what you're going to do, and why you're going to get back, and how to this isn't convincing other people that what you did was right. It's having them basically not disagree with you. They might not agree with you, but it's like, here's the math, and this is what it is, and this is how we draw conclusions from it based on the original mechanism plausibility. So it's another step in de-risking this brand new molecule I'm going to put inside of an animal or a person or a whatever, or a plant. It's saying, okay, at least we know what, this is the point. It's like, well, how do they know stuff? Like, this is how you know stuff. This is how people know what they know, because they go out and they do it. And then they end up with like a book like 5 inches thick that is the full description of how they did it and why they did it and all this other stuff. So moving on to non-cellular models, this is blood. This is stuff like, okay, I'm starting from the small and then getting larger here. So this could be something like blood. Does your drug interact with the blood? So you pull it, you ask, whatever, we're going to use butcher, like you go to a butcher or whatever. Not even like that, but like, if you're doing this right, you're going to have, a volunteer or a sample. There'll be blood. There'll be, you'll basically dose the blood to see if something happens. So you can either do whole blood or fractionated blood, but the idea is blood itself is not a cellular model. There are cells in the blood, but the blood itself is not the cell itself. You're not going into you're not looking at the DNA of the blood. You're not looking at the Golgi apparatus of the blood. You're looking at the blood, the whole blood. Did you coagulate the blood with your molecule? Yes or no? Did you react with the clotting factors in the blood? Did you coalesce the lipids in the blood? Did you just make a cholesterol clog with your molecule? It's that type of pre. So it's like, if you did, you got to go back to start. Wah, wah. Back to start. the same thing exists where it's like it's a non-cellular model. So blood, semen, hair, nails. It's more a little bit more relevant in the cosmetic section where it's like, well, if the keratinized layer of the nail gets corroded or weakened by the nail polish or the remover or the faux nail adhesive, go back to the, like, how bad is it, and then go back to the drawing board. Did the hair dye that you put in, denature all the proteins and ruin the hair? If it did, we could sell it as a hair removal device. That's where, that's where Nair comes from. That's where, like, that's sort of the world of this drug development where it's like, you found a bad effect, but the bad effect can be used someplace else. Those are the ideas. Those are two hands working together. But those are non-cellular models. Those are like structural models. Those are the same thing if you're in botany. It's like, well, did the application change the pH of the surrounding soil and ruin your plant tissue and kill off your plant tissue that way? So it's things like that that you're really like you're not even here. You're like, whoa, like we're gonna zoom out. And you're still at a frame. Like we zoomed out. We're still at a frame on this because that's how many like things get stacked and are still relevant. So it's very much like each discipline can still use it. So if you're doing botany, it's like, well, does my growth formula kill off the mycorrhizal fungi in the plant that would have made it, in the soil that would have made the plant grow more had I not even applied it yet? So those are sort of like non-cellular models that you're looking at, where like, or did it change the pH of the soil and kill off, it's you're getting into these like things that nobody thinks about. So then there's this. So the same kind of rule applies to subcellular models. So does your molecule react on contact? Does it lyse cells? Does it denature the proteins on the cell surface? Does it interact with things? So here's the cell. Things that are usually inside the cell. If the molecule gets into the cell, is it going to the DNA? So this is where like an Ames mutagenicity test comes in. Did you, is this thing going to cause cancer? So we only have two types of like worlds of models, and if this thing causes cancer, you're not going, it's not going near anything. You know what I mean? That being said, it's like, it interacts with DNA. Directly and negatively. Sometimes it could be an intercalation. It can, there's a whole bunch of different mechanisms, but then it's like, okay, well, a lot of times if you're doing this as a high throughput, you just, okay, we'll put it down, say it reacts with DNA, we don't know why, we don't know how, but shelf it for now. Shelf this molecule family. Don't even think about using that structure anymore. Go back to the drawing board and come up with another mechanism. Back to start. That's sort of what you learn and discover at the subcellular model level. It's like, well, I'm not going to put it in a person if it gives them cancer. You know what I mean? But this is where you're really looking for drug targets. This is subcellular is kind of like where you're really going to find your drug targets for the mechanism of plausibility. So it's like, does it affect mitochondria? Does it affect Does it affect the lysosomes? Does it affect, the picture of the cell? Does it affect any one of those? Like, does it affect the centrioles, the centromeres, whatever you want to call it? Like, what is your molecule doing inside of the cell? Because when you switch over to the cellular models now are now going to take into account, okay, well, those are the parts and components of a working cell. Now we're going to change the nature and expression of all of those different parts and components. So we're going to change the concentration of ribosomes in your Golgi apparatus to go from a hepatocyte to a skin cell. Or from a kidney cell to a lung cell. So that's sort of the like, okay, well now your cellular models are changing because you're changing the cell types. There's different concentrations of different targets expressed in different cell types. So you now have to sort of say, okay, well now my molecule how is my molecule going to affect all of these different cell types? Hopefully it's going to be clear sailing across the board. Nothing except for the pathology, and that's how sort of like bacteria work. That's why an antiviral was created so quickly, because it's like the antiviral is not necessarily the cell, the human cell, monkey cell, the other animal cell, the plant cell. That's sort of the focusing point there, where it's just like, okay, well, if the target isn't human, but the human interacts with the target, so it could be a fungus, it could be a bacteria, it could be, antivenom. Antivenom is kind of like this. You only need an antibody for the antivenom protein. So as long as the human doesn't express the antivenom itself, then you can go really fast into you can get a product out relatively quickly. And then there's tissue models, so you can have organs obtained from abattoirs to demonstrate kidney filtration of the drug and clearances of the drug. So you're getting these types of you're going very slowly, and we're not building a replicating human in 24 a replicating model of the digestive system in 24 hours. So that's our music change. So that's kind of why these are where and why and how they're contained on this wheel, to just help you understand what they are. It's like you have to do your own expansion of what a cellular model is and why you use it, and why it's so important to get your statistics lined up ahead of time, and why you want to use those statistical tests for what you're doing in your experiment at the cellular level or the tissue level or the translational study. So the tissue, so more into the tissue model, it straddles the in vitro and the in vivo world, because it's got to come from somewhere. So abattoirs, slaughterhouses are good places to start to understand how an organ is going to function in its physicality, not in its physiology, not in its biophysiology, but in its biophysics. So you can basically filter under pressure a kidney and see if this concentration of drug is going to cause kidney stones. You know what I mean? Like it's that type of thinking that you set up and design your trials for your new molecules with. And then you do a translational study, because basically you should have some good dosing information from all of your other non-living tests and systems. So if you're doing a new, and this is why new molecule development is so just fucking expensive, because you now have to take your new molecule and your new molecule, because your new molecule is kind of representing the new molecule family. So if you're a new molecular development, this is what drives you crazy, because you now have to go to the FDA and say okay, well, here's our tox screen. Here's our work with the non-cellular models, the subcellular models. The molecule has to pass every single one of these tests to prove safe and efficacious for use in the treatment of a disease. We're not even at the disease model yet. So, well, we are at the disease model. I'm gonna a little bit of backtracking here. Not to start, but these cellular models, you can choose and pick. So if you're doing Alzheimer's research, some of your non-cellular models can be the amyloid plaques or extracellular exudates of the amyloid plaque producing cells. So it's extremely specific to the disease itself. And that's why you always go back to start, because it's like, well, someone may have written a paper about like, oh, well, you know, this molecule kind of worked, which then saved you like, oh, I don't have to like, well, I can scrap 2 of my ideas and start with something that kind of worked, and then grow from that, and then grow or develop from there. So that's sort of the specifics. So what the hell is it? Which brings us back to a translational study. So what the hell is a translational study? So it's like, okay, well, the non-animal models tell you something like dosing information. So this is where you kind of have to sit down and basically say, if all of my previous results were in an animal, this is my expected outcome. And you have to, and this is more of like, you sit down, and it's another planning exercise, it's another planning study. It's more saying, it's like, okay, here's what I know about the animal I want to use. Here's what I know about the disease model that I want to use. Is my new heart medication going to screw over the kidneys? Because the heart and the kidneys are linked. It's part of the renin-angiotensin system. Also your lungs. So there's about 4 different organs that have to go into direct consideration, because they're sharing direct feedback loops with each other. So if you raise the blood pressure in one, but your kidneys stop working because you destroy the microvasculature, or you stopped urinating to increase blood pressure, and you know what I mean. Like there's these types of multivariate higher level considerations that have to go to absolutely everything. It's not enough that your car needs oil, but the amount of washer fluid in your car will directly affect the performance of your engine's output. That's how this kind of gets. That's why this kind of gets like really non-intuitive. It's not a linear system. It never was a linear system. It's like power based and logarithmic. So your translational study actually has to sit down. It's like here's my results, and here's where I want to go. Is there going to be problems with where I want to go? And the translational study can be short and sweet, where it's like, well, here's some dosing information, and here's what I know about the animal and the person, and based on some other studies or something, like there might not be any information. So you kind of just have to like, okay, well, you never actually wing it, but at the same time, it is like, here's this stuff, and then here's this thing that's alive, and they now have to interact. Based on the previous data, this is how I anticipate it to interact with something that's alive, fully alive, not tissue, not cell culture, not tissue subcultures or artificial organs or stuff like that. The artificial organs are in the tissue model, so it is really just sort of like, okay, well, here's the full deal of everything you know. How's it going to interact? It's like, okay, well, start the show. And again, again, your animal models here are, so we're not actually like, we haven't bought it, we haven't even gone, like, we haven't even purchased, like, there are no animals involved here at this point. It's like the animal models themselves are like, is your animal genetically, so for cancer, it's like, is your animal genetically engineered? Is your animal a wild type? So that means it's like, it's like, the mouse you buy in the pet store is a wild type. You know, there might be some genetic knowledge about it, but it's not cloned. It doesn't have any knockout genes associated with it. There's no silence genes. There hasn't been any modifications. It has an immune system. It is basically street rat. You grab a street rat and say, okay, from the street rat, all of these other different lines and outright species, almost species of mice and rats and other animals have been developed to act and serve as models for diseases. So there's like a cystic fibrosis mouse, there is a diabetes mouse, there is I was going to spina bifida mouse, but that's more of a surgical creation kind, I think. I'm not sure on that, so don't freak out. So it's like there are different mouse models, and which mouse is going to be appropriate for you. And even still, so even in the cancer world here, it's like, okay, here's 3 different like mice species now that are used routinely in cancer research, but the method of the application of the cancer cells, so you have to give the mouse cancer. So the mouse has to get cancer in some way, shape, or form, so you can treat the mouse with your new sort of disease solution. Or your plant has to grow or express its mosaic virus. You know, it's a disease, it's a pathological disease state. So you are looking at like, okay, well, what is the most relevant to the person in clinic? What is the most relevant to the orchard in the field? You want some control so you know if something goes wrong, or you don't see a lot of, you don't really want to use wild types because you are looking for something, usually something very specific. A lot of times this is done for the purposes of we want and or need to learn more about the underlying mechanism of the disease itself, and that's where most of those like species are used. But at the same time, you do have to keep it relevant to the wild type population kind of thing. So there's always that plausibility of give and take, and this is why your in vivo work is 1, 2, 3, 4 is so 4 out of the 6 sections on the wheel there are just planning sections. So then your animal models, and then you got to go with ethics approval. For the most part of drug assessment, there really isn't any ethics. It's like you're gonna administer the drug to the animal, and that's it. That in and of itself is a standardized protocol. Sometimes it requires a sedative. Sometimes it requires, something else. Like if it's a surgery, you have to do an anesthesia. You have to have control groups. You have to have things like this that ensure that the one thing that you're doing is actually just the one thing that you're doing. There's no confounding. There's no voodoo or hoodoo in the mix kind of thing. It is just like, okay, this treatment works because I did exactly this. Full stop. That's the ethics approval. Plus, you have to get it approved by, university, third parties. There's an ethics board for use of animals at some point. If you're in any type of facility that isn't like floating in international waters, that basically says the standard of care is done to approximate the people involved that you will be helping. So it's like, do people eat every day? So are your animals fed? People clean themselves every day. They also have access to water and litter change. Like, you know what I mean? So put the person in the cage that the mouse is going to be living in. Put yourself in the cage. You know, put yourself in the cage that the mouse is going to be living in, and then make sure your mouse is held to that standard. Because there's a lot of animal psychology stuff here where it's just like, well, if you had to like walk on your shit every day and trapped in a box, well, you'd be absolutely fucking miserable too. So you have to have that type of stuff planned ahead of schedule and written down. Also, you have to be able to demonstrate that the application of the thing is not like just, this is not just a sadistic experiment. You have to say like this is for a purpose. And you have to demonstrate that it's not just like, the experiment itself is not overtly sadistic. Well, we're gonna chop the head off the mice and then give it some drugs. Like, no, no, that doesn't, like, no, that's not getting approved. That's just like, fuck off. Like, you know, like that's the part where you're just like, no, go, like you're just being, nobody will ever, like, nobody who's serious about, like, no serious money will ever just be like, yeah, okay, no, go, you know, go to a barn, go step on a mouse in a barn someplace. So once you get your ethics approval that your experiment and what you're proposing is actually going to be valuable and worthwhile to the people who the mice represent. Because you don't want to go into the clinic and do this. You want to do this on, like, you want to get all the information without being like the data for the sake of data type of people here. You have to get this right. This is an oversight in getting things right. And then on top of the ethics, so that's just for like what you physically plan to do. But then there's the statistical power assessments, and this is like one of those things. Going back to the bioassay, way back in vitro work, it's like the statistical power assessments are making sure that you have enough data points, and it could be animals, but it could be sub-animals. It's really, this is like where you're getting scrutinized in terms of your experimental designs and what you want to have done here. And that's basically where you have to have X number of mice to be able to minimalistically draw a certain set of conclusions for what you want to do. So if you do this on one mouse, it's like, okay, like don't get me wrong. There are things that we're over time. It's gonna be one of those hour and a half, 2 hour ones. Statistical. The power assessments here are basically, it's like, is there enough statistical power to be able to validate the study design that you chose? If you like, and it doesn't mean that you're gonna have one mouse. One mouse could still satisfy everything. But if you're doing one of these like drug trials that are like, okay, well, we're just gonna do like 50 trials, like the treatment and the control group and the ancillary treatment groups all have to have a sufficient number of mice in them. Because what if one dies sooner or later? So you need to be able to have an exclusion, a group large enough to exclude the outliers or rare and random events associated with your treatment protocols to basically draw any valuable conclusions to the number of people that would. So it's like one drug to cure all the people with the disease. That's kind of where this kind of comes from. So it's like, if you're going to use this one molecule to this one drug to cure everybody with cystic fibrosis, and there's like a couple million people with cystic fibrosis, but you're only using 10 mice, 10 genetically engineered mice, that does not make sense. You have to have like 100 mice here for that type of statistical power for that one molecule. There's ways around this type of stuff, but this is like make your life easier, go through that hell first, and then set up the so that you just have the analysis computations, so that you just have to enter your own self-derived data points into it. And then animal husbandry. So that kind of goes back to be the mouse in the cage, basically. Pretend to be the mouse in the bed, in the cage. Pretend to be the person in the bed. If you don't actually have the disease yourself, that's kind of like the best idea about how this type of work actually goes. I use animal husbandry here because it's like you still haven't actually with a cancer model. It's like the day you inoculate, it could be like a month that these animals grow the cancer or like have the cancer growing before you actually do anything. So this is kind of how all this sort of gets put together, where it's just like no, this is actually like no, this is all pre-planned. So like you have to have the cages set up, you have to have people there, you have to have the food, the water, the linens, the cleaning, not linens, but you have to have the sheets, or not the sheets even. You have to have those little curly things, the wood chips, right? You have to have the little wood chips there up front and ready for when you start this. That's part of the ethics approval. That's part of like how you're setting this up. That's part of your entire in vitro set that you have to set up yourself, basically, and oversee and control for your one molecule, because we know what the clinic looks like, and this is the clinic for animals. Unfortunately, we have to create the animals with the disease to help the people who get the disease on their own. So that's kind of what the prescription, that's what in vivo looks like when you're hearing like animal drug trials and why they get so expensive. Like there's so much thinking and pre-planning into this, it's ridiculous. That was the industrial model that was set up in the 50s. By now, there are multiple companies that keep breeding stock of mice, and that's what they do. They professionally, this is the more, this is tumor farm. That's tumor farming, basically. In my world, it's tumor farming. That's what the tumor farming looks like. This massive industry grew up and sprouted for different disease production things, and it's you know, I like the sun, it's called pineapple, but that's the whole idea. It's like, okay, it's a farm. It's a place where people go. You can grow the mice. Sometimes they raise some pets. It's a regular sort of like pet shop. It's a pet shop. That's what it is. It's a pet shop. For specific purposes. And nothing's been done to the animal. So it's like the next step is treatment data. So here's your treatment. So in your animal husbandry phase, this is where you do your actual treatment. It's like you have to put the mice or the rats in the if you're taking blood from it, you do the veil thing stuff. This is where the action happens. This is tumor farm. All of it's tumor farm. It's all tumor. If you have to milk the rat bladders, this is where it happens. If you need rat urine to see if the drug is passing through unchanged or if it's being excreted at all. So milking rat bladders for urine is one of the animal husbandry types of jobs. So anyway, treatment data and analysis. So you do what you did to get these numbers. And hopefully, because there have been 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 to, I don't know, maybe we're at 18-ish type of oversights that the animal lives were worth it. So, treatment data and analysis. If everything goes right, you actually got dosing information to go right to clinic with. The animal animals lived, you cured their disease, huzzah, huzzah, hooray. And now you have a molecule with information you can actually use on a human being. And then he's like, back to start. So you go back and you just sort of repeat the process over and over again. And you don't have to pick a new molecule, but you do have to do your stat and your you have to do your model, you have to do your ethics approval, you have to do your statistical power assessment, and all over again before you actually recruit people for the actual agent administration. So the study's design approval is basically, is this like, if we do this, if we use this drug at this concentration relative to the person's body size, we will get the same results you got in mice. There is you save a lot of work, but you still have to do it again for people. It's its own thing. People are the sacred cow here. So whatever you did in the animal, you can use a lot of the basic structure for that, but you have to then redo it all for the people. And you get study recruitment thrown on top of that. So study recruitment says it's like, okay, so the molecule might work, but you are obligated to work with the specialist doctors and the specialist pharmacists. At this point, at this point, this is all still one company, because these experts are the other people assisting you through this process. They're not your general physicians. They might be your specialists in the hospital, but from the level I'm talking at, they're actually working for the tumor farm level company here. So what they're doing is looking at, it's like, okay, if this molecule compound is used in a person and the same sort of side effects are observed, so they're looking for side effects from the animal study and sort of like projecting that into the people, and then looking at further saying, okay, well, some things might not be observed in people that were observed in the mice, and some people, so they're very much like they're veterinary with human, they're like MD, veterinary and human doctor, they're MDs with DMVs, like at the same time kind of thing, because there's like these interspecies differences. They're translational specialists. So assuming everything sort of signs off on that, now you got to go find a whole bunch of doctors to be able to help to administer it. So study recruitment is basically, it starts at one site with low number of people. If a human protein or something interacts with your molecule that wasn't caught way back in your in vitro sides, because you can use human proteins in your non-cellular and subcellular stuff, and there's recombinant stuff. That's the sort of idea here. So it's like you just can't use humans in your in vivo work, but you can use human stuff in your in vitro work. So that's kind of what goes on. You can kind of start with humans now, anyway, and then sort of artificial human, and then go through. And there's no reason why you can't go back and use more in silico along the way. But the idea here is like, we know a lot about what the human being is going to do now, and there's tons of databases for it, plus textbooks, plus university programs, plus all of this. Is like there's no real sort of Anyway, study recruitment. So you start with like small number of people, because you're into the regular doctors do no harm type of thing. The study will get shut down immediately if something goes wrong. It's shut down immediately. it's basically looked at as like an emergency situation. We've entered in, like, clinically, this is an emergency situation. It's end of life or something like that. I'm speaking from more of like where I'm coming from, but if it's an antibiotic, it's a new antibiotic, it's like, well, they're gonna die anyway, so you know, you are like legally obligated almost to use this new thing on them because it is like a compassionate care or an emergency situation where nothing else is known to exist. So we've tried 4 rounds of antibiotics and you still have this infection. Can we have permission to use this new research molecule? Yeah, like they're like, oh my god, yes. But that's sort of the thing where it's like you get, and this is sort of doled out through the hospitals, but you have to go from your synthesis pathway all the way to your study recruitment to make sure that your doctors have, you know, what is now a formulated medicine to give to the study patients. You have, you know, now so you like the difficult part in prescription development is you have to set up a manufacturer supply line at the same time as you're running to be able to fund, and not only fund but be able to feed into all of the experiments that you have to do to help the people you want to help. It's like you build the factory first, and that's what makes drug development so extremely expensive. You build the factory first, and then you try and find, you know, where this thing works. But the thing is, it's like, it's not just one molecule that you've, you know, standardized for. We're not just making rolls of tape here over and over and over and over again. We're turning the tape into toilet paper and to paper towels and to wrapping paper and to basically any other type of tree-based product you can stick on a roll. That's sort of like the real frustrating and expense of it, where it's like, no, it's a synthetic organic laboratory that's able to handle just about everything. And then the difficulty there is that you want to add a molecule, you want to then, you know, you might have a reaction in the human portion of it, and this is what goes from the clinic back to in silico, where it's like you might have something good in the human, like there might be symptom relief, but it's not doing what you wanted, so you have to go back to your functional group screening type of stuff in silico and say, okay, it's kind of doing what I want, but it picked up a different organ system, but if we change this functional group or, you know, lop it off or make it bigger, it'll get, you know, it's too big to, like, if the normalized receptor goes like this, but if we make it big enough to do this, if we make it big enough to do this instead of this, then it won't be in that organ system long enough, and therefore it won't be able to cause as many downstream effects. So it's like if you have a receptor binding, it has a lower affinity for a different organ or organ system than it does for the target site and its organ system. You understand what I mean? It's just like, oh my god, it just keeps going on like this. So like it doesn't, like, you can have a really positive result. You got symptom amelioration from this, but it caused some other stuff that wasn't that great. So you could go back and try and redesign it, because you do have like a lot of information, but you don't necessarily have to do the whole thing again. You have to test the like you don't have to do the whole, like, what I mean by you don't have to do the whole thing again is like, if this analog is behaving the same way in your cellular models, you don't have to use as many animals, because you already have confirmation this is the behavior pattern of the class of molecules. So this class of molecules is going to behave roughly and approximately the same way. So instead of using 100 mice, you can use like, you use 25 or 50, just to say, it's like, is the toxicological profile or the pharmacological profile the same at the same dosages with this modification, yes or no? That's how you develop drugs. That's how you not be stupid with data for the sake of data. so that you can go back to the clinic and say, okay, well, we got some symptom relief with this. We didn't see any additional side effects. We didn't see any surprises. We got exactly the same results with this one as we did with the mice. We'd like permission to go further, and then you leave it up to the ethics board kind of thing, because there's still like nothing. But anyway, so study recruitment, you got some doctors, you got some patients that are, clear, level-headed, and willing to agree. And then treatment administration. So you do what you did with the mice. You don't have to do what you do with the mice because the people understand. So they'll sit for 3 hours, or watch TV or something while they have an intravenous drip, or they're not going to squirm and kick the needle out. Basically, that's sort of the idea here. It's like, or it might be a pill, where they can just take it themselves. Treatment administration, but that's another problem where it's like, well, if you end up with adherence. They take all the medication that was prescribed because of a side effect, or because they forgot to take it with food. So there's all that kind of fun stuff. So all that kind of like wonky zonky stuff goes into the study design approval. So we're giving out antibiotics to people, and they didn't take the full prescribed amount, and they got reinfected because they didn't take this prescribed amount. So it's like, well, expand the trial then, and like duplicate another trial set to make sure that they take all the drug, like take all the doses. So that there's all that type of stuff too that is like drives you crazy, because it all affects your data analysis. So if you got your study design and you got your statistics set up at your study design level, the data analysis allows you could be clear sailing here. And this sort of sells like this is the point where like you're gonna have to sell it. It's gonna either sell itself at this point, or it's going to have to go back to start for additional behind the scenes like industrial development. So that's the information reporting stage. So like, yay, it passed the stats. Now write up everything you did in like English language, so that one, you get a copy of the book. Two, you have something to read. And then if you're in a different nation, you can send this to, like, if you have like, so that you can now disseminate that information and change the disease profile to being something like, okay, well, before we didn't have any treatment options for it, but now we have one. So that changes the disease profile in the sense that it's no longer a terminal disease. It is now a chronic disease. It is now no longer a chronic disease. It is an acute disease. It's now no longer an acute disease. It is a curable disease. So you can sort of backtrack and go through all of this stuff and understand just like, oh my god, how many times did you go back to start? And it's like, so what Innovative Potential did here is we did the in silico work, we did the portion of the in vitro work, because we're using a molecule that has completed the full cycle. So cyclophosphamide has been through this cycle multiple times for about 50 years now. So it's been through this once, and then it was sort of backfilled. So the molecule in question has each one of these sort of things really well described, thanks to 50 years worth of grad student and university and NIH, Health Canada type of work done in their own laboratory. So that's how and why a lot of the information, I was like, well, if we take a cellular model on how this is behaving, and if it's matching the cellular model, we can kind of jump to a human model, because we know the molecule itself is not we know its toxicity, we know how the drug works we know how the molecule works as a drug, so we can skip the in vivo stage and go to study design and approval at the human stage. That's how that works. That's the idea behind innovative potential, where it's like, well, we already know the in silico because the drug model itself is already complete. We're using a completed drug model, not once but twice, for the device, because the device is a thing that's been tested, not the drugs. You know how they work. So this wheel is completely filled for both acetaminophen and cyclophosphamide, and most of their analogs. So the functional group screening and the analog profile is like fully completed for this wheel for like 3 or 4. There's like 4 to 11 of these wheels fully filled out for each of the drugs that are compatible with the Joey. There's a mountain of evidence that says the Joey works. What has to be done is the finalizing these 3 in the blue section: the study design, the study recruitment, the treatment administration, data analysis for the specific device itself per drug. So it's not insurmountable, but it does kind of take your in-clinic drug and put it back in the clinic. So basically the Joey only functions in clinic because it doesn't, but has markets available in the in vivo and in vitro and in silico sections that basically help you design new classes of drugs using the mechanism specific to the Joey. So that's the electrochemical modification, reduction oxidation of the molecule itself. So basically what the Joey is an in vitro artificial subcellular model for in vitro screening, but it's also a device for treatment administration. How neat is that, right? So that's the real power behind innovative potential and some of the stuff we've done, and the electrochemical generation of reactive species and their application as chemotherapeutics. That's what that book allows to be done, where you're kind of living in a world without it. And that's it for the prescription development. That is the requirement for FDA approval if you start with a brand new molecule. If you that sort of thing. Well, why isn't the FDA on here? Why aren't the regulators on here? Blah, 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 blah. It's like the information reporting section. It's the academic. It's more from the scientist perspective than it is from the regulator perspective, but it satisfies both of them. If you have this much information, yeah, they're both satisfied, kind of thing. And they are both satisfied. It's just sometimes it's a matter of money or a matter of publicity. One way or the other, doesn't matter. But the point is, the drug and how it works, and the information that is being reported on has the rest of this entire wheel backing it up as to why it works. That's de-risking. Like, that's what this is. But it's like, oh, well, how did, then you get one of these things, well, how did Vioxx happen, where it was like, oh, it was working good for like 50 years, but was found to actually cause heart attacks. And it's just like, well, part of that, and it goes into the goes into how many people it actually goes into, because it's like, if you want to have one drug go through 24 million prescribed patients, you cannot ask a company to be directly responsible for testing that many people. That's a number that goes, if you go into our big number video, it's sort of like that number comes out at a geography or continent level of resolution. A company can't do a whole geography. That's 8 million people directly samples. There are drug monitoring programs But if the demography of your geography changes or is not amenable to a specific drug, or is, so yeah, so if a drug works better in black people than it does in white people, then it's also just as wrong to not have that drug prescribed more in that geography because there's more black people in that geography, or vice versa, or South Americans or Australians or whatever. That's sort of how like different people's biology works. It's like what it is. And pretty much it. So this is the wheel of prescription development. It's the one I got stuck on, just because I couldn't figure out how I wanted this to work in relation to the other four wheels. This is sort of there's just sort of the everything on it. This is like a wheel of asset creation. Asset creation is the general one. This is more very specific to innovative potential and how something like the Joey is able to take your drug and basically get it to clinic faster. The Joey here is functioning as a functional group screen because it's acting as a subcellular model independently of the enzymes within the cell, and is able to produce good parallels and approximations to actual metabolic processes outside of the body and outside of the cell. There are some things to be aware of, and we discussed that in the book. Like the lipophilic environment of the Golgi apparatus would produce different metabolites than the aqueous environment of the Joey. That depending on what your endpoint is there, that can be adjusted. The effluent of the Joey can be adjusted. The composition of the Joey effluent can be adjusted to make more lipophilic or less aqueous sort of effluents based on formulation. So you still got to have a small, depending on your drug, on your new formulation, on how that works, depending on the amount of information of your drug, that's where you can do a translational studies to say, okay, well, we've been using bleomycin forever. It's an anti-cancer drug that satisfies the redox activation of the Joey. If we just put it if we just take its formulation and run it through and do an intratumoral injection with it, I haven't tested it. I haven't used it. It's one of the it fits. It's the proper currency value for the vending machine, basically, right? Is it actually going to behave like it's actually going to behave through the full thing? It's like, well, if it's behaving if it behaves the way in silico and it behaves the way in vitro, and you have your in vivo models demonstrating some of that the active thing is still, the active molecule is still present in your in vivo data analysis, so your histological stuff or your blood or your urine samples, but you're able to verify that something approximating that is coming out of the effluent of the Joey without actually having to use the animal models. It's like if this is a roll of tape and this is a roll of tape, then the same roll of tape and the same roll of tape is used in clinic, well, then we can use bleomycin and the Joey in clinic. We don't have to do additional testing. It's like, oh, but what if one of these other things comes? Like, the whole body is designed for detoxification of everything. If you have a drug approved for intravenous administration, you basically have a drug approved for the Joey and intratumor injection. So that's sort of the generic world. You can patent different formulations and go to market with the Joey for this type of stuff. If you find a formulation that works better specifically with the Joey as effluent, then that would be, because it's like if you have a stabilizer in your formulation that would then deactivate the active compound, but would get your compound through the digestive system, let's say, or have it be more stable to survive phase one. I don't know. I was going to say phase one, but this is phase one metabolism. So it's like the idea here is like your formulation might actually obstruct the active molecule as an effluent from the Joey. So there might be some tweaking to your formulations, but you can patent the formulation. So that's how you make a panacea with the Holy Grail. We're done. That's out. We're done. That's the world of innovative potential. I still love that.